What first made you interested in transcription?
Um, that was when I was an undergraduate student, basically, and got a lot of lectures on molecular biology and really enjoyed um, understanding more about it and this sort of what you could call machines on genes type things, and got me fascinated, and that's how I got interested in studying gene regulation and the machinery that's involved in transcribing genes. And so that's how I got into transcription, which is that process that makes RNA from DNA.
Expect to find, were there any surprises?
So if it relates specifically to the study, yes, then yes, there were a few surprises because initially as I mentioned earlier, we had one PhD student, my first PhD student, who basically made a few mutations in residues that we thought were very important in coordinating zinc ions in this protein. And it totally killed the whole activity of this protein complex, not just that protein, but the whole protein complex didn't function. So that was a surprising result. And then we dug deeper and deeper and found that the protein was still nicely incorporated into the complex. The complex still bound DNA. That was still a surprise as well, because we thought initially zinc fingers. We thought if it's a zinc coordinating amino acids, then if we mutate those, we thought we might mutate a zinc finger and therefore not have DNA binding. But it wasn't a zinc finger. It turned out to be a slightly different variant that also is involved in coordinating zinc ions, but it had nothing to do with DNA binding. So that was a surprise, and then we dug deeper and then found that even the recruitment of the polymerase wasn't affected. So all in all, the mutations gave a very dramatic phenotype, and yet um, the we couldn't pin it down to any of the obvious defects in assembly of the complex, recruitment of the polymerase, DNA binding. They were all fine. And it turned out that actually there was a defect after all these events happened. So it binds DNA fine as a properly assembled complex. It recruits the polymerase, but then there is a problem. And that was actually also what led to what is a major hurdle in our analysis. It was to actually then prove we knew where the defect wasn't, but we didn't know where the defect was exactly, and to prove that was actually very difficult.